This figure shows cardiomyocyte which are connected to each other by intercalated discs. The cardiomyocytes have a central nucleus, plenty of energy supplying mitochondria, and muscle fibers. They also have a rich capillary blood vessel network. This figure shows dark areas crossing the cardiac muscle fiber cells. These are known as intercalated discs. They are cell membranes separating individual cardiac muscle cells from one another. These have various electrical channels to exchange ions like sodium and potassium and strong adhesion areas like desmosomes. They have collagen fibers and blood vessels in between the cells. Dear listeners, this video is part 2 of the 4-part series on anatomy and physiology of heart under the Prescribon medical education series. You have already seen the structure of cardiac muscle and the intercalated disc. The dark areas crossing the cardiac muscle fibers are called intercalated discs. They are actually cell membranes that separate individual cardiac muscle cells from one another. That is, cardiac muscle fibers are made up of many individual cells connected in series and in parallel with one another. At each intercalated disc, the cell membranes fuse with one another to form permeable communicating junctions, that is, gap junctions, that allow rapid diffusion of ions. Therefore, from a functional point of view, ions move with ease in the intracellular fluid along the longitudinal axis of the cardiac muscle fibers so that action potentials travel easily from one cardiac muscle cell to the next, past the intercalated discs. Thus, cardiac muscle is a syncytium of many heart muscle cells. Note, syncytium is a single cell or cytoplasmic mass containing several nuclei formed by fusion of cells or by division of nuclei. The cardiac cells are so interconnected that when one cell becomes excited, the action potential rapidly spreads to all of them. Note, action potential. A rapid rise and subsequent fall in voltage or membrane potential across a cellular membrane. The heart actually is composed of two syncytia. The atrial syncytium, which constitutes the walls of the two atria, and the ventricular syncytium, which constitutes the walls of the two ventricles. The atria are separated from the ventricles by fibrous tissue that surrounds the atrioventricular AV valvular openings between the atria and the ventricles. Normally, electrical potentials are not conducted from the atrial syncytium into the ventricular syncytium directly through this fibrous tissue. Instead, they are only conducted by way of a specialized conductive system called the AV bundle, a bundle of conductive fibers several millimeters in diameter. See later. This division of the muscle of the heart into two functional syncytia allows the atria to contract a short time ahead of ventricular contraction, which is important for the effectiveness of heart pumping. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. This figure shows a cardiomyocyte, and these myocytes are connected to each other by intercalated discs. They have a central nucleus and plenty of energy supplying mitochondria. They have a rich capillary blood vessel network. This figure is a figure of a cardiac myocyte, which shows cardiac myocytes with thick and thin filaments, which is the contractile apparatus, sarcolemma, which is the cell membrane of a myocyte, the Sarcoplasmic Reticulum network, the terminal cisternae, and the T tubules, and the mitochondria. The cardiac myocyte. See the earlier figures of cardiac myocyte and the cardiac muscle cell. Cardiac muscle cell is striated like skeletal muscle, as the actin and myosin are arranged in sarcomeres, just as in skeletal muscle. However, cardiac muscle is involuntary. Cardiac muscle usually has a central nucleus. The cells are often branched and are tightly connected by special junctions. The cell-to-cell connections are known as intercalated discs, situated at 90 degrees to the striped sarcomeric pattern. The intercalated discs contain gap junctions, adherence junctions, and desmosomes. Gap junctions, large and small, provide direct contact between the cardiac muscle cells, allowing them to be electrically connected so that waves of depolarization spreads rapidly over the entire heart by passing from cell to cell so that they beat in synchrony. Adherence junctions are strong mechanical junctions between the two cells. Actin filaments attach their filaments to the cell membrane. Desmosomes are the site of strong anchoring junctions between the two cells. The ventricular myocyte is a large, elongated cell 100 to 150 micrometer long and 20 to 35 micrometer wide, and is packed with striated myofibrils, these contractile elements that alternate with rows of mitochondria. Sarcolemma is the cell membrane of a myocyte. The myocyte is composed of bundles of myofibrils that contain myofilaments. The myofibrils have distinct repeating microanatomical units termed sarcomeres, which represent the basic contractile units of the myocyte. The sarcomere is defined as the region of myofilament structures between two Z lines. The distance between Z lines, that is the sarcomere length, ranges from about 1.6 to 2.2 mu, that is 1 mu is 1,000th of a millimeter. Each myofibril is roughly cylindrical, 2 to 3 micrometer in diameter, stretches the length of the cell, and is anchored at each end. The myofibril comprises sarcomeres arranged in series. This is the picture of actin and myosin. Shown is myosin filament and one head of it having ATP, that is energy processing element. Shown below is the unit containing actin filament colored red, tropomyosin, troponin C and troponin I units, which will be explained below. This picture shows the picture of a sarcomere. The thin filament actin is blue colored, the thick filament myosin is red colored. Shown also are the sarcomere unit, M line, I band and the A band, which will be explained shortly. Sarcomeres consist of two arrays of filaments. Thin filaments comprised predominantly of the protein actin, interdigitated with thick filaments of myosin. The characteristic striated appearance arises from the organization of these myofilaments within the myofibril. The thick filaments are confined to the A band at the center of the sarcomere. The thin filaments extend out from either side of the Z disc, that is the Z line, crossing the I band, and penetrate partially into the A band, where they overlap and interact with the thick filaments. Each Z to Z disc repeat constitutes a sarcomere, and the distance between consecutive Z discs, the sarcomere length, is a measure of the contractile state of the myofibril. At the center of the sarcomere lies the M line. Each myofibril contains 70 to 80 sarcomeres. Myocytes have an irregular branched morphology. Through these branches, each ventricular myocyte typically connects to 10 or more of its neighbors to form the three-dimensional branching syncytium-like structure of the myofiber. Sarcoplasmic Reticulum, SR, is a specialized membrane-bound endoplasmic reticulum, is a network of tubules storing calcium ions. Usual concentration is 10,000 times less than the concentration of calcium ions outside the cell. The reticulum network extends throughout muscle cells, wrapping around but not in direct contact with myofibrils. Transverse tubules, otherwise known as T-tubules, are extensions of cell membrane that travel into the center of the cell. T-tubules are closely associated with a specific region of the sarcoplasmic reticulum known as terminal cisternae, which are situated at every 12 nanometers. A nanometer is 1,000th of a micrometer. This is the primary site of calcium release. The M line contains the protein myosin that cross-links the thick filaments to maintain their orientation. Recall the structure of actin and myosin again. Shown is the myosin filament in the upper part of the picture, and one head of it is a structural arrangement which releases the energy processing element called ATP, adenosine triphosphate. Shown below is the unit containing actin filament, red colored, tropomyosin, and troponin C and troponin I units, which are explained herewith. Myosin is a protein having a molecular weight of approximately 470,000 daltons. There are about 300 molecules of myosin per thick filament. Each myosin contains two heads that are the site of the myosin ATPase, an enzyme that hydrolyzes ATP, that is adenosine triphosphate, required for actin and myosin cross-bridge formation. These heads interact with a binding site on actin. The thin filaments are composed of three different types of protein: actin, tropomyosin, and troponin. Together, these are termed the regulatory protein complex. The actin is a globular protein arranged as a chain of repeating units, forming two strands of an alpha helix. Interdigitated between the actin strands are rod-shaped proteins termed tropomyosin. There are 6 to 7 actin molecules per tropomyosin. Attached to the tropomyosin at regular intervals is the troponin complex, which is made up of three subunits: troponin T, TNT, which attaches to the tropomyosin; troponin C, that is TNC, which serves as a binding site for calcium during excitation contraction coupling. 4 calcium ions can bind per TNC, and troponin I, TNI, which inhibits the myosin binding site on the actin. When calcium binds to TNC, there is a conformational change in the troponin complex such that TNI moves away from the myosin binding site on the actin, thereby making it accessible to the myosin head. When calcium is removed from the TNC, the troponin complex resumes its inactivated position, thereby inhibiting myosin-actin binding. TNI is important in clinical practice because it is used as a diagnostic marker for myocardial infarction, because it is released into the circulation when myocytes die. Changes in sarcomere length are an important mechanism by which the heart regulates its force of contraction. As a myocyte is stretched, as occurs with increased ventricular preload, the sarcomeres within the myofibrils are also stretched. Note, ventricular preload is the load of blood which stretches the heart cavity and hence the muscle fibers before muscle contraction. With increased sarcomere length, there is an increase in the force of contraction, that is, tension development by the muscle fiber. For many years, it was thought that the major mechanism for the increased force generation was an increase in the overlap between actin and myosin, which made more sites available for hydrolysis of ATP, that is adenosine triphosphate, by the myosin ATPase. This was an application of the sliding filament theory as described for striated skeletal muscle. Sarcomere lengths do not change very much in cardiac muscle compared to skeletal muscle. Nevertheless, small changes in sarcomere length can produce large changes in tension development. It is now known that stretching the sarcomere increases TNC affinity for calcium, which leads to increased tension development. This is referred to as length-dependent activation. For various structural and mechanical reasons, the sarcomere length in the cardiac myocytes does not normally exceed 2.2 mu. Shown here is the diagram of coronary circulation which supplies the heart. From the aorta, the right and the left coronary arteries arise. The RCA, that is the right coronary artery, supplies right side of the heart, and the LCA, that is left coronary artery, supplies the left side of the heart. The left coronary artery has an anterior descending artery and the left circumflex artery. More details follow. Now we will see the coronary blood flow in detail. There are two primary coronary arteries, the right coronary artery RCA and the left main coronary artery LMCA. Both of these originate from the beginning of the aorta. The right coronary artery supplies blood primarily to the right atrium, right ventricle, SA node, that is sinoatrial node, and the AV node, atrioventricular node. The right coronary artery then descends into smaller branches, including the right posterior descending artery, PDA, and the acute marginal artery. The posterior descending artery supplies the posterior 1/3 of the interventricular septum, IVS. The left anterior descending artery supplies the anterior 2/3 of the septum. The left anterior descending artery is one of two major branches of the left main coronary artery, with the other being the left circumflex LCX coronary arteries. Combined, these two supply blood to the left atrium and left ventricle. The circumflex artery is responsible for blood supply to the left atrium and the posterior lateral aspect of the left ventricle. Left anterior descending artery supplies blood to the anterior portion of the left ventricle. Other small branches of the coronary arteries are the obtuse marginal artery OMA, diagonals, and septal perforator SP. The limitation of blood flow through the coronary arteries that cause malfunctioning of the myocardium is known as coronary artery disease, CAD. Stenosis of the branches of the LMCA or the RCA affects specific locations of the heart. Complete occlusion of the left anterior descending artery results in an anterior wall myocardial infarction. Occlusion of the left circumflex results in a lateral wall MI. Occlusion of the posterior descending artery results in an inferior infarction. Dear listeners, this is the end of part 2 in this 4-part series. For other videos, see Prescribon Medical Education Series Anatomy and Physiology of Heart.